Welcome to the Inner Ascent, a journey beyond the surface. Each week, we distill powerful insights in mindset, health, personal evolution, and conscious growth. Unlock the layers of your mind, one ascent at a time.
The content of this podcast is for informational purposes only and does not constitute medical advice, diagnosis, or treatment. Always seek the advice of your physician or a qualified health provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay in seeking it because of something you heard in this podcast. The views expressed are those of the speakers and do not necessarily reflect official positions. Use of any information provided is solely at your own risk.
Welcome to the Deep Dive, your shortcut to being truly well-informed. Today, we're diving deep into mesenchymal stromal cells, MSCs. We'll look at what they are, how they're being explored for really complex diseases like multiple sclerosis, and their exciting, maybe even surprising potential to combat the effects of aging.
Yeah, it's a fascinating area. And for this deep dive, we've pulled together insights from, well, quite a range of sources. There's a scientific review focusing on MSCs for MS, a big scoping review on anti-aging and stem cells, even a perspective from a commercial clinic talking about what they call high-potency MSCs.
Right, and we also looked at a report on the costs of going abroad for these therapies and a specific study using an MSC product for diabetes-related cognitive issues.
Exactly, so quite a mix.
Our mission really is to unpack the science. What are these cells doing? What are the different ways they might work therapeutically? We want to explore the current landscape, treatments, challenges, all of that, and, highlight some maybe unexpected links between these cells and our long-term health.
The goal is you walk away with the key insights. You'll be genuinely well-informed on what's happening in this cutting-edge field.
Okay, let's start by setting the stage. Maybe look at some of the big health challenges MSCs are being aimed at. First up, multiple sclerosis, MS.
Right, MS. It's a chronic illness, and it targets the central nervous system. It's basically an autoimmune condition. It's inflammatory, and it's also neurodegenerative. The body's own immune cells, T cells specifically, they attack the myelin sheath.
That's the protective coating on nerve fibers.
Precisely. And when that gets damaged, you get neurological problems, nerve fiber damage, what's called axonal loss. It's really debilitating for many people.
And the current treatments, disease-modifying drugs or DMDs?
Yeah, the DMDs. They can definitely help. They can reduce how often attacks happen, how severe they are, and protect the CNS a bit. But, and this is a big but, they have limits. They don't really repair existing damage like axon repair, and they often come with significant side effects.
So there's a real gap, a need for something new, something maybe regenerative.
Exactly. That's where the interest in therapies like MSCs comes in. There's a clear need for better options.
Okay, and then the other big area, aging, something that affects literally everyone. We think of it as just, birthdays piling up, chronological age.
But it's much more than just the number of years. Aging fundamentally is about the time-related deterioration. Things just not working as well. Physiological functions you need for survival, for fertility, it happens in all organisms.
And you mentioned this crucial difference, chronological versus biological age.
Yes, that's key. Biological age isn't just time passed. It's a whole spectrum. Your physical function, physiological state, how sharp your mind is, and it's really driven by things happening at the molecular and cellular level.
Things we can actually see and feel, right? Vision changes, hearing loss.
Strength decline, bone density dropping, immune system not as robust, maybe some cognitive fog, less energy.
Hair loss, moving slower. The list goes on.
It does. But underlying all those visible signs are these fundamental cellular and molecular processes, the hallmarks of aging.
Hallmarks of aging, like what specifically?
Well, things like genomic instability, DNA damage accumulating, telomere attrition, those protective caps on chromosomes getting shorter every time a cell divides.
Until the cell just stops dividing, senescence.
Exactly. Cellular senescence.
Yeah.
The cell kind of retires, stops dividing, but sometimes these senescent cells hang around and cause problems, like inflammation. And that's another hallmark, chronic low-grade inflammation.
So these underlying mechanisms are what are really driving the aging process we experience.
That's the idea. And if we can target those mechanisms, maybe we can influence biological aging.
Which brings us perfectly to mesenchymal stromal cells, MSCs. You hear them called the body's inner healers. It sounds powerful, but what are they, fundamentally? What makes them special?
Okay, so MSCs, they're multipotent stromal cells. Not blood-forming cells, that's important, non-hematopoietic. They're kind of spindle-shaped, and they can self-renew, make more of themselves.
And they have specific identifiers. You mentioned markers.
Yeah, that's crucial for identifying them properly in the lab. They stick to plastic in culture. That's one characteristic. And they express certain surface markers, proteins on their surface. Positive for CD73, CD90, CD105.
Positive for those.
But negative for hematopoietic markers, the ones found on blood cells. These markers are like their ID tags. Helps ensure you're working with the right cell type.
And they can turn into other cell types, differentiate.
In the lab, yes. They can be coaxed to become chondrocytes, cartilage cells, osteoblasts, bone cells, and adipocytes, fat cells. That's the multipotent part.
What seems really appealing is getting hold of them. You said they're quite accessible.
Relatively speaking, yes.
Yeah.
Compared to some other stem cell types, they're easier to harvest and culture, grow in the lab, and they tend to maintain their genomic stability when expanded, which is important for safety.
And you can get them from lots of places.
Bone marrow is a classic source, but also adipose tissue fat. And perinatal tissues are becoming really important. Umbilical cord, placenta, Wharton's jelly.
Wharton's jelly. That's the stuff inside the umbilical cord.
Exactly. It's rich in these MSCs.
Okay, so accessible, identifiable, can differentiate. But you also called them natural pharmacies. What's the mechanism there?
Right. The natural pharmacy idea comes from how they mainly work. It's not always about becoming a new tissue cell themselves. Their biggest impact is often through paracrine signaling.
Paracrine, meaning they release substances that affect nearby cells.
Precisely. They secrete this complex cocktail of bioactive molecules. It's called the secretum.
The secretome. That's an N.
All sorts of things. Growth factors, cytokines, which are signaling molecules for the immune system, and tiny little packages called extracellular vesicles, like exosomes.
Exosomes. Those are getting a lot of attention lately.
They are. They can carry signals directly into other cells.
Right.
So the MSC doesn't need direct contact. It releases these factors, and they go out and influence the surrounding environment, calm inflammation, promote repair, act like a pharmacy delivering specific instructions.
So it's less about becoming the new brick on the wall and more about being the foreman directing the construction crew and putting out fires.
That's a great analogy, actually. Yes. They orchestrate responses.
So this orchestration, how does it work with the immune system? You mentioned immunomodulation.
That's one of their strongest suits. They are potent immunomodulators. They can suppress excessive inflammation, which, as we said, is a big problem in aging and in autoimmune diseases like MS.
How do they calm things down? What are they telling the immune cells?
They interact with lots of immune cells. They can stop T cells and B cells, key players in adaptive immunity, from proliferating too much or maturing improperly. They also affect antigen-presenting cells like dendritic cells and macrophages. They can stop them from maturing in a way that ramps up inflammation and even push macrophages towards an anti-inflammatory state.
So they're like diplomats negotiating peace.
Kinda, yeah. They release anti-inflammatory signals like certain cytokines. IL-10 is a key one, maybe IL-6 in certain contexts. And they inhibit pro-inflammatory cytokines like TNF-alpha, which is a major driver of inflammation. It's about restoring balance or immune homeostasis.
Okay, immune calming. What about actual tissue repair and neuroprotection?
Well, they can contribute directly by differentiating, as we mentioned, replacing damaged cells. But again, the signaling is huge, especially for neuroprotection.
In the brain and nervous system.
Yeah. Studies, often in animal models first, show that if you inject MSCs, say intravenously, they can reduce the activation of microglia, those brain immune cells, which can cause damage if overactive. They can protect neurons from dying, promote remyelination, getting those oligodendrocytes to repair the myelin sheath damaged in MS, and they can prevent too much scarring from astrocytes.
And they produce helpful factors, too.
Definitely. They can nudge nearby glial cells to produce neurotrophic factors, things that support neuron survival and growth, like NGF, nerve growth factor, BDNF, brain-derived neurotrophic factor, VEGF, vascular endothelial growth factor.
Oh, okay, so they really are multitaskers. How does all this connect back to the broader idea of anti-aging?
It connects directly to those hallmarks we talked about. Mitigating chronic inflammation check, dealing with oxidative stress check, modulating cellular senescence. They seem to influence that too. They can enhance mitochondrial function, the cell's powerhouses, help remodel the extracellular matrix, which gives tissues structure and elasticity, promote angiogenesis, new blood vessel formation for better nutrient delivery, and even stimulate autophagy, the cell's cleanup process for damaged parts.
So unlike a drug that might target one specific pathway, MSCs seem to hit multiple aging hallmarks at once.
That's the potential, yes. It's a more holistic approach, addressing the complex interplay of factors involved in aging, rather than just one isolated mechanism.
It helps to see this in a specific case. Let's talk about that diabetes study. Diabetes increases the risk of cognitive problems, right? Linked to the hippocampus.
Yes, significantly. High blood sugar can lead to oxidative stress, inflammation, cell death, particularly damaging the hippocampus, which is vital for memory.
And this study used something called WJMSC-CM.
Right. Wharton's jelly mesenchymal stem cell conditioned medium. So this is key. It's cell-free. They grow the Wharton's jelly MSCs in the lab, collect the liquid medium they were grown in, that's the conditioned medium, full of the secretome we talked about.
Ah, so it contains all those beneficial factors, the growth factors, cytokines, exosomes.
Exactly. But not the cells themselves. They filter the cells out. Then they use this WJMSC-CM to treat diabetic rats.
Why cell-free? Is that an advantage?
It can be. It potentially reduces risks associated with transplanting whole cells, like immune rejection, even though MSCs are relatively immune privileged. And it might be easier to standardize and store as a product.
Okay, so what happened in these diabetic rats treated with the conditioned medium?
The results were pretty striking. The treatment helped improve their high blood sugar, first off. But importantly, it significantly improved their cognitive function and reduced anxiety-like behaviors.
And at the cellular level in the brain.
It reduced oxidative stress markers, lowered neuroinflammation, specifically bringing down TNF-alpha levels, and decreased neuronal apoptosis, meaning fewer brain cells were dying in the hippocampus. It also helped preserve the size, the volume of the hippocampus, and actually increase the number of neurons and other supportive cells like astrocytes and oligodendrocytes.
Wow. So the secreted factors alone were enough to have these protective and regenerative effects.
That's what the study suggests. The benefits were attributed to key growth factors found in the CM, like HGF, FGF2, BDNF, NGF, VEGF, and also the exosomes. These factors, especially in exosomes, might even be able to cross the blood-brain barrier to act directly in the brain.
So this points towards a way to treat something like diabetes-induced cognitive decline by targeting the root causes inflammation, cell death, using just the therapeutic payload of the MSCs.
It's a really promising avenue, yeah. Using the power of the cell's pharmacy without the cell itself.
Now, when we talk about actual MSC therapy using cells, a big practical point is where they come from. Autologous versus allogeneic. Can you break that down?
Sure. Autologous means the cells come from your own body. They harvest them from you, maybe bone marrow, maybe fat tissue, then typically expand them in a lab and give them back to you.
And allogeneic?
Allogeneic means the cells come from a donor, a different person, usually a healthy screen donor. Often, these are the perinatal sources we mentioned, umbilical cord, placenta, Wharton's jelly.
What are the downsides of using your own cells autologous, especially thinking about aging?
Well, the harvesting itself can be invasive. Bone marrow aspiration isn't pleasant. Adipose tissue extraction is surgery. And sometimes you just don't get enough cells. The yield is low. Plus, and this is really relevant for anti-aging, the number and the quality, the differentiation potential, the potency of your own MSCs tend to decline as you get older. Your aging cells might not be the best medicine for your aging body.
Ouch. So using your own older cells might be less effective.
That's a concern, yes, which makes allogeneic sources look very attractive.
Why are they often preferred now?
Several reasons. You skip the invasive harvesting from the patient. You don't have to wait weeks for the cells to be cultured and expanded. They can be available off the shelf, so to speak. They can be isolated in much larger quantities initially. They generally have low immunogenicity, meaning less risk of rejection. They don't trigger a strong immune response. And crucially, their potency isn't tied to the recipient's age. Plus, using perinatal tissues often raises fewer ethical objections than, say, embryonic stem cells.
So among these donor sources, are some considered better than others?
Perinatal tissues are generally favored now. Umbilical cord, Wharton's jelly, placenta. They're considered rich sources of young, potent MSCs.
You mentioned Wharton's jelly MSCs, WJMSCs, specifically before?
Yes, WJMSCs are getting a lot of attention. They're seen as having a more primitive nature, good multi-lineage potential. They reproduce quickly in culture. Animal studies have shown promise, like in MS models, where they seem to reduce demyelation and help regenerate the myelin sheath.
And placental or umbilical cord MSCs.
Also very promising. Abundant sources, easy to access after birth, and they also show strong immunosuppressive capabilities. There's a lot of research ongoing with all these perinatal sources.
This all sounds incredibly hopeful, but it can't be all smooth sailing. What are the risks or challenges scientists are still working through?
Absolutely. It's not without challenges. There are potential risks. Some studies have raised concerns about fibrosis or scarring. Sometimes, depending on the context, MSCs might actually promote inflammation temporarily, a pro-inflammatory effect. And a major concern that's always carefully monitored is tumorigenicity, the potential for cells to form tumors. That's considered a leading risk factor to rule out in clinical applications.
Wait, so they could potentially cause cancer or help existing cancers grow?
It's complex. Some studies suggest MSCs might suppress tumors. Others suggest they could potentially promote tumor growth or angiogenesis, the blood supply to tumors under certain conditions. It seems to depend heavily on the specific context, the type of cancer, the state of the MSCs. It's an area of very active research and caution.
And you mentioned variability in results earlier.
Yes, that's a big hurdle for clinical translation. Why does it work well for one person or in one study but not another? It could depend on so many things.
Like?
The dose, how many cells are given, how many times they've been passaged or divided in the lab. Too many passages might reduce potency. The way they're injected via IV, direct injection, the timing early in the disease versus late, and the patient's own specific disease state. Lots of variables.
This complexity and maybe the slow pace of regulatory approval in some places leads to this whole phenomenon of stem cell tourism, doesn't it?
It does. You mentioned the FDA restricting Wharton's jelly MSC treatments in the U.S. around 2019, citing lack of proven efficacy from large trials. That's a significant factor.
So people feel they can't wait, or the cost is too high for experimental trials at home.
Exactly. High costs, limited access to approved trials, sometimes just desperation. It drives people to look elsewhere, to countries with perhaps different, maybe more permissive regulations. It's sometimes called regulatory arbitrage, seeking treatments abroad that aren't approved or available locally.
Where do people typically go? You mentioned Colombia.
Colombia is one destination, yes. Clinics like BioAccelerator operate there, promoting advanced protocols, selecting specific potent cells they call signature cells. Panama, Costa Rica, Mexico are also common. And we saw testimonials mentioning places like Turkey for MSC. It's a global market.
It creates such a difficult situation. You have these patient stories, sometimes dramatic testimonials.
People reporting significant improvements, brain fog lifting, sleep better, less pain, even unexpected things like hair regrowth. And there's really striking, though often anecdotal, reports about spinal cord injury patients regaining some function. Those stories offer immense hope.
But then you have the scientific community urging caution.
Right, because outside of rigorously controlled clinical trials, it's very hard to prove definitive efficacy. How much is placebo effect? How much is the natural course of the disease? Are the reported benefits truly due to the stem cells?
And concerns about the clinics themselves. Quality control.
That's a major concern. Are patients getting what they pay for? Is it a high-quality preparation of viable, potent MSCs or something much less? Sometimes cynically referred to as sugar water. What about the cell quantity, the passage number? Some clinics might use cells expanded many times, maybe to passage 7, raising those questions about potency.
There are also debates about how they're administered, right? Like 5V for joint problems.
Yes, the efficacy of simple IV infusions for orthopedic issues is questionable for some experts. The concern is that most intravenously injected MSCs get trapped in the lungs quite quickly, the pulmonary first pass effect. They might not reach the target joint in sufficient numbers.
So for joints, direct injection is better.
Generally preferred, yes. But even then, there are concerns about how it's done. Is it guided by imaging, like ultrasound or fluoroscopy, to ensure the cells get to the exact right spot? Or are they blind injections, which might miss the target and be less effective, possibly less safe?
Okay, let's try to wrap this up. It's a lot to take in. We've covered the basics of MSCs, these potential inner healers.
Their multipotent nature, those key characteristics.
And their mechanisms calming the immune system, helping with tissue repair, neuroprotection, mainly through that complex signaling, the secretome.
Right, acting like those natural pharmacies.
We've seen how this applies to tough challenges like MS, potentially slowing aging by hitting those hallmarks, and even that specific example of fighting cognitive decline in diabetes using the cell-free conditioned medium.
And the shift towards allogeneic sources, especially perinatal tissues like Wharton's jelly, because of the advantages over using potentially older, less potent autologous cells.
But we also touched on the real-world complexities, the risks, the variability, the regulatory hurdles leading to stem cell tourism.
And the critical need to balance those compelling patient stories and hope with rigorous scientific evidence from proper clinical trials, ensuring safety and quality control.
So looking at the big picture, MSCs are undeniably an exciting frontier in regenerative medicine. They offer this really multifaceted approach, but it leaves us with a big question, doesn't it?
Definitely. It's the central tension, perhaps.
As these advanced therapies keep developing, how do we strike that balance between the desperate hope of patients needing solutions now and the absolute necessity of robust science, careful ethics, proper oversight to make sure treatments are genuinely safe and effective for everyone? What really stands out to you thinking about the future here, especially for tackling aging and chronic disease?
You've been listening to The Inner Ascent, where growth begins from within. If today's reflection resonated with you, consider sharing it or revisiting it later. Until next time, stay aware, stay curious, and keep rising.